Welcome back to the Deep Dive, the place where we take the vast, overwhelming ocean of research and distill it down to the essential, fascinating knowledge you need.
Hey, everyone.
Today, we are grappling with one of humanity's greatest achievements and, simultaneously, one of its greatest self-inflicted crises, the story of antimicrobials.
That's right. When we look back at the 20th century, the discovery and deployment of antibiotics, well, it stands out as arguably the most successful form of chemotherapy ever devised.
Just incredible drugs.
They really are miracle drugs. The lives they saved are, they're in the uncountable millions. They transformed surgery, public health, basic medicine forever.
Yeah.
But that very success has led us to a critical junction, a point where the incredible evolutionary power of the microbial world is now, well, it's catching up,
Right.
threatening to undo a century of medical progress through the emergence of these hard-to-treat, multi-drug resistant infections.
So our mission today, using the stack of sources we have in front of us, is a deep dive into this whole complex arc. We're unpacking the history, the ecology of resistance, trying to understand not just, how penicillin was discovered, but maybe why resistance is this ancient, ongoing evolutionary problem.
And what complex, multi-layered strategic efforts are needed, like right now, to sustain the efficacy of these really foundational drugs for generations to come. It's a huge task.
It sounds enormous.
And we really have to frame this for you, the listener, with the urgency it demands. This isn't some, distant theoretical thing.
No.
It's an immediate global crisis with escalating human and economic costs. Just to put a number on the human toll, in the European Union alone, approximately 25,000 patients die every year.
25,000.
Every year.
Yeah.
From infections caused by selected multi-drug resistant bacteria. That's like a medium-sized town it just wiped out annually, purely because our chemical arsenal is failing.
That's a staggering statistic. And the economic footprint, I imagine, is just as bad.
Just as staggering. Places massive burdens on healthcare systems, global productivity. Our sources detail that the estimated economic costs in the EU, and this is factoring in extra healthcare costs, lost productivity from sickness, death, it's at least 1.5 billion euros.
Billion? What, a B?
Every single year. It's like a persistent draining tax on society.
And the U.S. picture.
It mirrors this perfectly.
Yeah.
It just shows how global this is. Our sources, using 2006 dollars, mind you, show the annual extra cost of treating hospital-acquired infections from just 6 highly dangerous resistant species.
Just 6 species.
Yeah, was already estimated at over $1.87 billion.
Back in 2006.
Exactly. And that number has only climbed since then, probably exponentially. The problem is entrenched, it's incredibly expensive, and it's accelerating faster than our ability to innovate, really.
Okay, let's unpack this. Before we can even think about tackling the crisis, we need to completely sort of dismantle a foundational misconception, one that really limits our thinking. The idea that our exposure to antibiotics is strictly a modern thing, confined to the age of labs and chemistry and drug discovery in the 20th century. It's wrong, isn't it?
Completely wrong. We need to go much, much further back to when humans first started really interacting with the natural microbial world.
Oh.
Yeah. Challenging that timeline, the start of the antibiotic era, is maybe the most critical first step to understanding the current resistance crisis.
Okay.
The evidence is just overwhelming. The 20th century didn't start the fight. It just gave us the chemical tools to amplify a biological interaction that's been happening for millennia.
Humans, microbes, antimicrobial compounds, they've been co-evolving for a very, very long time.
And this delicate, ongoing balance, absolutely.
And the most tangible proof, the really compelling stuff, it comes not from a lab initially, but from archaeology, right? This discovery of, accidental tetracycline consumption thousands of years ago.
It's one of the most stunning pieces of evidence in this whole deep dive, honestly.
Right.
Researchers found chemical traces of tetracycline, a powerful antibiotic class we still use, in human skeletal remains.
Where from?
From ancient Sudanese Nubia. And these date all the way back to 350 to 550 CE.
Wow. So 1,500 years ago.
Exactly. And tetracyclines have this unique chemical property. They are strong chelators. Meaning they bind really tightly to metal ions, especially calcium.
Okay, and why is that binding property so crucial when you're looking at bone?
Because bones and tooth enamel are mostly made of this calcium phosphate mineral called hydroxyapatite.
Right.
So when tetracycline is ingested, it circulates in the blood and gets permanently incorporated into the newly forming hydroxyapatite matrix during bone mineralization.
Ah, so it gets locked in as the bone grows or repairs itself?
Precisely. It essentially leaves a permanent, unmistakable chemical marker, it's called a fluorochrome label, in the parts of the skeleton and teeth that were metabolically active, forming or remodeling during the time of exposure.
So finding it in the bone structure, not just on the surface, that's the smoking gun, proving they actually consumed it repeatedly.
That's exactly it. The specific, consistent way that tetracycline is distributed within the bones can only be explained by consistent long-term exposure through their diet.
Through their diet. How?
Well, the most likely source, though we can't be 100% certain, was probably consuming fermented grains or maybe even beer that was contaminated with naturally occurring bacteria that produced tetracycline.
Like streptomyces, those soil bacteria.
Exactly, strains of streptomyces. So they were unknowingly consuming this medicine as part of their food.
And this wasn't just an isolated thing in Nubia, was it? You mentioned other finds.
No, it wasn't a local fluke. A
Hmm.
histological study, actually done years before the Nubian find, looked at late Roman period skeletons from the Dakhla oasis in Egypt. Same story. They found discrete fluorochrome labeling patterns consistent with tetracycline presence. Again, it points to their diet, including this antimicrobial compound. It suggests these natural antibiotics might have been pretty widespread contaminants in certain historical food processing systems across huge geographic areas.
Okay, so they consumed it. What's the aha moment here? Is it just interesting or did it actually do something? Did this ancient low-dose exposure have any clinical effect?
Well, this is where it gets really interesting. Our source material points to a strong possibility that it actually provided a protective effect.
Like a low-level preventative medicine.
Essentially, yes. They might have been benefiting from a continuous low-dose prophylactic treatment without even knowing it. For example, the documented rate of infectious diseases in that Sudanese Nubian population, it was noted as being quite low.
Huh.
And researchers noted no traces of systemic bone infections, things like osteomyelitis, in those Dakhla oasis samples either. So they may literally have been healthier because their beer was contaminated.
That is a stunning thought. Accidental contamination being a good thing, potentially.
A hidden protective factor, yeah. But exposure wasn't just accidental food contamination. Traditional and alternative medicines played a documented role too, didn't they?
Absolutely. Traditional Chinese medicine, TCM, gives us maybe the clearest historical path for humans deliberately encountering antimicrobial substances.
Right, and exerting that long-term, low-level selective pressure on microbes. Using herbal remedies provided these substances long before modern pharmacology was even a concept.
And the rediscovery of artemisinin is the perfect example of that, isn't it? Ancient knowledge meets modern science.
It's the definitive case study. Qincao, which we now call artemisinin, is this incredibly potent anti-malarial drug, crucial globally today.
Yeah.
It was chemically extracted and purified back in the 1970s from Artemisia plants. But here's the thing. Chinese herbalists have been using these very plants as remedies, often for fevers, malaria-like symptoms, for thousands of years.
So it wasn't a brand new discovery, more like a quantification of ancient knowledge?
Exactly. It was ancient knowledge just waiting to be systematically analyzed and purified. And we know antimicrobial activity is present in hundreds of other TCM herbs as well. It's a vast natural pharmacy.
So the selective pressure wasn't just industrial, starting in the 20th century. It was built into culture itself through medicine for millennia.
That's the critical insight here. The long-term history of using these natural antimicrobial remedies, both accidentally in diet and intentionally in medicine, could easily have been one of the factors contributing to the slow, steady accumulation and maintenance of antibiotic resistance genes within human-associated microbial populations way back in historical time.
It created a sort of pre-selection environment.
Precisely.
Okay, this brings us to the truly massive time scale of the problem, evolution. If resistance was around in 500 CE, how far back does it really go? Are we talking thousands of years or something much older?
We need to shift gears from history to deep, deep geological time. Phylogenetic reconstruction, mapping gene evolution based on genetic changes over eons. It confirms the long-term presence of resistance genes in nature, well before humans were before we were using antibiotics in any significant way.
The microbial world has been innovating for billions of years.
Eons.
Can we get specific? Which resistance genes are we talking about, and what does the evolutionary clock say?
Well, let's look at the beta-lactamases. These are the enzymes bacteria use as their primary defense against things like penicillins and cephalosporins. They're like chemical scissors that destroy the drug.
Okay.
Structure-based phylogeny suggests that both major classes of these enzymes, the serine and the middle lactamases, they originated over 2 billion years ago.
2 billion with a B.
Yes. 2 billion years ago. To put that in perspective, complex multicellular life like animals only showed up maybe 600 million years ago. These resistance mechanisms are ancient microbial survival tools.
That makes our 100-year antibiotic struggle seem like a blink of an eye, really.
It absolutely does. And furthermore, if you look at the genes in a common hospital pathogen like Klebsiella oxytoca, the analysis of its beta-lactamase genes alongside its regular housekeeping genes shows such similarity in evolutionary path.
Meaning?
It implies these resistance genes have been co-evolving within that specific bacterial lineage for over 100 million years.
Wow.
They didn't just pop up in the 1950s when we started using penicillin widely. They were already there waiting.
And even looking at more recent history, geologically speaking, the data still points to ancient origins in the environment.
Exactly. There's metagenomic analysis of 10,000-year-old deep-sea sediments, places completely untouched by modern human activity.
Okay.
And it shows that the incredible diversity of these resistance enzymes we see today is the result of ancient evolution. This diversity was already established in the environmental resistem, that's the natural collection of all resistance genes out there, long before we started mass-producing penicillin.
So the microbial world was, in a way, ready for us.
You could definitely say that. Prepared.
Okay, here's where it gets really interesting for me. If resistance is billions of years old and microbes have been bumping into low levels of antimicrobials for millennia, what finally clicked? What kicked off the modern systematic antibiotic era just over a century ago? What changed to bring these tools out of the soil and the lab bench and into the clinic in such a massive way?
Well, the shift wasn't just finding a molecule. It was really about establishing a conceptual framework.
Okay.
The idea that you could systematically engineer a chemical agent to treat an internal disease. That concept gave the organizational principle, the systematic approach needed to isolate, synthesize, and then deploy these powerful compounds reliably.
And the father of that concept, that's Paul Ehrlich, right? With his idea of the magic bullet.
Ehrlich's genius, starting in the early 1900s, was realizing that synthetic chemicals could potentially be designed to selectively target disease-causing microbes without harming the host cell.
What inspired that?
He was initially inspired by the emerging synthetic dye industry. He noticed certain dyes would stain specific microbes, but leave the host tissues untouched.
Ah, suggesting a biochemical difference you could exploit.
Exactly, a difference in affinity. He formulated this principle that compounds could be synthesized that would, in his words, be able to exert their full action exclusively on the parasite harbored within the organism.
Which led him to set up the first really systematic, almost industrial-scale drug screening program in history.
Yes, starting around 1904. His huge program aimed squarely at finding a treatment for syphilis.
Which was a terrifying disease back then.
Absolutely devastating. Caused by the spirochete Trypina pallidum, the standard treatment involved highly toxic inorganic mercury salts. It barely worked and often killed the patient anyway. Ehrlich wanted something truly selective.
And after years of just relentless systematic testing with his colleagues Bersheim and Hatta.
6 years, yeah. They finally hit the breakthrough, compound 606, an organoarsenic derivative. It successfully cured syphilis in infected rabbits and showed immediate, really significant promise in human trials. It was marketed as Salvarsan in 1910.
Salvarsan.
And it became immediate global success because, frankly, despite its own toxicity issues, it was dramatically more effective than anything else available.
It's kind of incredible to think that this arsenic-based drug was the most frequently prescribed drug, along with its successor, Neosalvarsan, for decades, wasn't it? Until penicillin finally took over in the 1940s.
It really was the first true success story of modern chemotherapy. It defined the start of the era.
But here's a fascinating detail from the sources. Despite being used on millions, they didn't actually know how it worked for a very long time.
That's right. It's amazing. Its precise mode of action and even its exact chemical structure remained controversial, even kind of unknown, for almost 100 years.
A century, while it was the top drug.
Yeah. It turns out the drug needed to be chemically oxidized inside the body to become active against the spirochete. That was a subtlety scientists couldn't fully unravel until much later with modern analytical techniques.
Wow, that really highlights the difference between that early empirical success, it works, let's use it, and our modern expectation of knowing the exact molecular target.
A stark contrast, absolutely. Treating millions successfully for 40 years without fundamentally knowing the how at a molecular level, it just shows the urgency of the medical need back then.
Definitely. But Ehrlich gave us the concept and the synthetic approach. We should also mention the biological precursors, the hints that were already there in nature.
Yes, absolutely. You're thinking of the sort of unsung precursor, piocyanase.
Exactly. Tell us about that.
Well, unknown to many. The first time a drug we'd now call an antibiotic was used in a hospital setting was actually piocyanase. It was prepared by Emmerich and Lowe way back in 1899.
From what?
From the bacterium Pseudomonas aeruginosa. They noticed these extracts were active against several other pathogenic bacteria, and they actually tried using it for various diseases, including infections in children.
But it didn't really take off, did it? It was abandoned pretty quickly.
It fell out of favor because the results were really inconsistent. The potency varied wildly from batch to batch, and the preparation itself turned out to be quite toxic to human tissue.
Okay.
However, later research absolutely confirmed that P. aeruginosa does produce antibiotic substances. We now understand these are mainly signaling molecules, part of his quorum sensing system.
Quorum sensing. That's how bacteria kind of count themselves, right? To coordinate actions.
Exactly. When the bacteria reach a high population density, they release these molecules specifically, a family called 2-alkyl-4-quinolones, in this case, to trigger group behaviors, like producing antibacterial compounds.
So the original idea of using natural extracts was right, but Emmerich and Lowe were working with this crude, complex cocktail of signaling molecules, not a purified weapon.
Precisely. And that early failure really underscores why the chemical precision achieved later with penicillin was so necessary for success.
Which brings us, inevitably, to the most famous story of them all, penicillin. The ultimate tale of serendipity meeting prepared mind, starting in 1928.
Yeah, the initial observation by Alexander Fleming, September 3rd, 1928, of that penicillium mold inhibiting bacteria on his agar plate that's foundational.
The famous contaminated plate.
Right. But Fleming's true contribution wasn't just noticing it. It was his formidable persistency, his belief in the idea. For 12 years, he kept trying to get chemists interested in solving the really difficult purification and stability problems.
He kept the flame alive.
He really did. And he freely supplied his penicillium strain to anyone who asked for it during that time. Plus, his screening method using those inhibition zones on agar plates to see the killing power that was relatively simple, resource-like.
And it became the standard.
It became the standardized technique that researchers in industry relied on for mass screening and testing potency later on. Hugely important.
But Fleming himself eventually kind of gave up on the clinical side around 1940.
He did. It was the Oxford team, led by Howard Florey and Ernst Chain, who took on the absolutely enormous challenge of chemical purification.
And they succeeded.
Their work was instrumental. By 1940, they published the paper describing the purification of enough penicillin for actual clinical testing. Their protocol, then refined massively in partnership with U.S. and U.K. industrial chemists during the war.
That led to mass production.
Yes. By 1945, penicillin was being mass-produced and distributed. It fundamentally changed casualty rates in World War II and basically revolutionized medicine in the decades that followed.
And yet, what's so remarkable and maybe often overlooked in the triumph is Fleming's almost prophetic hindsight about the danger lurking beneath the surface.
He was startlingly prescient, wasn't he? Fleming was among the very first to issue a stark warning about the potential for resistance. He cautioned it could emerge if penicillin was, in his words, used too little or for a too short period during treatment.
He saw it coming. He anticipated the evolutionary pressure and the bacterial counter-response decades before the resistance crisis really hit us globally.
Quite remarkable foresight.
Okay, so alongside the mold discovery path, we also have the synthetic pioneer path. Sulfanilamide, derived from Prontosil. This represents the purely chemical approach.
Yes, sulfanilamide defines another key foundational route, kind of bridging Ehrlich's synthetic work with the Golden Age. Prontosil was synthesized by chemists at Bayer, but it was just a precursor, a prodrug.
The body had to activate it.
Right. The liver metabolized Prontosil into the active drug, which was sulfanilamide. Now, the crucial detail here is that sulfanilamide itself wasn't new. It was already being used in the dye industry.
Ah, so it was off-patent.
Exactly. Off-patent and cheap to produce. This allowed pharmaceutical companies everywhere to immediately jump into mass production and start making chemical modifications.
And this oldest class of synthetic antibiotics, the sulfur drugs, they unfortunately provided one of the earliest and most widespread examples of resistance, really setting the stage for the modern crisis.
It was almost catastrophically effective at promoting resistance. Sulfur drug resistance is almost universally linked with these mobile genetic elements called class 1 integrons.
Integrons. Explain those again. They're like docking stations for genes.
That's a great analogy. They are highly mobile genetic platforms that are incredibly good at capturing, integrating, and then disseminating multiple resistance genes all at once, often across different species of bacteria.
So a bacterium gets one of these integrons, maybe on a plasmid, and boom, it's resistant to several things simultaneously.
Precisely. And what's particularly insidious about the sulfa drug resistance genes is this. Once that resistance mechanism gets established on a mobile element like a class 1 integron, the whole package, the integron plus the resistance genes it carries, seems to actually confer a measurable fitness advantage to the host bacterium.
Even when the drug isn't around.
Even in the complete absence of antibiotic selection pressure. It literally pays, evolutionarily speaking, for the bacteria to carry that resistance package around, whether or not the sulfa drug is present in its environment.
That is terrifying. It sets up this self-perpetuating feedback loop. The resistance doesn't just fade away when you stop using the drug.
Not necessarily, no.
So these early years, Salvarsan, piocyanase, penicillin, sulfanilamide, they define the paradigms of treatment, absolutely, but they also, maybe unknowingly, define the inevitable arms race that we are now so deeply locked into.
They absolutely did. The systematic screening from Ehrlich, the purification breakthroughs from Florian Chain, the synthetic route with sulfa drugs, they ushered in the golden era of discovery.
An era of stunning achievement.
But one that ultimately couldn't be sustained at that pace.
Right. The early years, say the 1950s through the 70s, that was the golden era of discovery. But then the well just ran dry, didn't it? Just as resistance really started to accelerate. That golden era, spanning roughly 20 years, maybe a bit more, between the 50s and 70s, it was an explosion of pharmaceutical innovation. New classes of antibiotics seem to be popping up almost constantly.
Aminoglycosides, cephalosporins, macrolides, tetracyclines, quinolones. It was an incredible period.
But our source material is pretty stark here. That golden era ended. And critically, no truly new classes of antibiotics have been discovered since then.
It's a pharmaceutical drought that has now lasted for, well, close to 50 years.
50 years with no fundamentally new types of antibiotics.
Broadly speaking, yes. The simplest way to think about it is that the low-hanging fruit was picked first.
Okay.
The easiest places to find new classes, particularly those well-known soil bacteria like the Actinomyces, they were just exhausted by extensive screening efforts using the methods available at the time.
So, with the pipeline for novel discoveries drying up, what became the main strategy?
The primary strategy shifted to chemically modifying the existing proven antibiotic classes, creating slightly tweaked derivatives of penicillin or cephalosporins or macrolides.
We're essentially trying to stay one step ahead by making the bullet, I don't know, a little faster or a slightly different shape.
That's a good way to put it. But we know the inevitable lesson learned from that strategy, don't we? Bacteria are just far better and faster at evolving than we are at synthesizing tweaks.
Right.
Sooner or later, bacteria will acquire resistance to these modified versions as well. It happens either through horizontally acquiring entirely new resistance mechanisms from that vast environmental resistom we talked about, or through rapid molecular evolution of the resistance mechanisms they already have to counter the older versions of the drug. They just adapt what they've got.
We're running on this rapidly accelerating treadmill, and the bacteria are consistently gaining ground.
That's the reality we face.
There is one really striking, kind of fascinating anomaly in this whole resistance story, though, that our sources highlight. The bug that causes syphilis.
Ah, yes, Treponema pallidum. It is a scientific mystery, or at least a powerful exception that proves the rule, perhaps.
Why is it such an exception?
Well, it's surprising because while resistance just exploded in common pathogens like the Enterobacteriaceae, gut bacteria like E. coli and Klebsiella, that became resistant not just to penicillin, but even to our last resort, drugs like carbapenems.
Yeah.
The organism causing syphilis, Treponema pallidum, somehow still remains highly susceptible to penicillin.
Which is astounding, considering penicillin became widespread in the 1940s. That's over 80 years of exposure for this pathogen. Why hasn't evolution caught up here?
Researchers speculate it comes down to the organism's unique biology and genetics. T. pallidum has an extremely limited genome compared to other bacteria.
Okay.
It cannot survive outside a human host, and it replicates very, very slowly. Most importantly, it seems to lack the mobile genetic elements, the plasmids and integrons we mentioned earlier, that allow for rapid horizontal gene transfer between bacteria.
So it can't easily pick up resistance genes from other bugs.
That seems to be the key. Resistance often relies on acquiring foreign DNA, and T. pallidum appears genetically constrained from doing that effectively. It really shows that the path to resistance isn't universal. It depends heavily on the specific biology of the pathogen itself.
Okay, so syphilis is the exception. For pretty much every other major pathogen, though, we can classify exactly how they defeat our drugs. This brings us back to the classical mechanisms of resistance, the traditional bullet target model.
Right, this model essentially looks at two main strategies bacteria use to win the fight. Either they protect the target that the drug is designed to hit, or they actively defeat the bullet itself, neutralizing the drug before it can do damage.
Okay, let's break down protecting the target first. How do bacteria do that?
There are basically four key ways at the molecular level. First, simple modification by mutation.
Example.
Okay, the drug rifampin works by binding to bacterial RNA polymerase, the enzyme that reads DNA to make RNA. If a bacterium gets a random mutation in the gene for RNA polymerase, the enzyme changes shape just enough.
And the drug can't bind anymore.
Exactly. Can't bind effectively, resistance achieved.
Okay, second way.
Modification by enzyme. This is a bit more subtle. Think about macrolides, like erythromycin. Bacteria can produce an enzyme that chemically modifies a specific spot on their own 23S ribosomal RNA.
The ribosome being the protein factory.
Right. This tiny chemical modification prevents the macrolide drug from docking onto the ribosome, essentially making the whole protein factory insensitive to the drug.
Clever. Third mechanism.
Replacement. Instead of changing the target, they just swap it out for a resistant version or protect it. A great example is tetracycline resistance again. Bacteria can produce specialized ribosomal protection proteins.
What do those do?
They physically bind near the tetracycline docking site on the ribosome and essentially shoulder the drug molecule out of the way, shielding the target.
Like a bodyguard for the ribosome?
Kind of, yeah. And finally, there's protection at a more cellular or even population level, creating a physical barrier.
That's biofilms, right?
Exactly. Bacteria secrete massive amounts of protective sugars, called exopolysaccharides, to create this slimy matrix. This matrix acts like a physical shield.
How?
It slows down the diffusion of the antibiotic, preventing it from reaching lethal concentrations for the cells embedded deep inside. It's an external defense strategy for the whole community.
Okay, so that's protecting the target. Now the second broad category.
Yeah.
How does the bacterium defeat the bullet, neutralize the drug entirely?
Again, three main strategies here. One is modification of the drug itself, chemically altering the drug so it loses its punch. A classic example is adding an acetyl group to aminoglycoside antibiotics, which inactivates them.
Okay, second.
Destruction. This is the most famous one. Producing those lactamases, those powerful enzymes we talked about earlier that act like molecular scissors.
Nipping the key part of penicillin and related drugs.
Precisely. They hydrolyze and destroy the crucial lectin and ring structure, rendering the antibiotic useless.
And the third strategy for defeating the bullet.
Simple eviction, pumping the drug out. These are the efflux pump mechanism.
The bilge pumps.
Exactly. Think of them as tiny pumps embedded in the bacterial cell membrane or wall. They actively recognize the antibiotic molecule and pump it out of the cell faster than it can accumulate inside to reach a killing concentration.
So they just lower the dose inside the cell?
Effectively, yes. Keeping the internal concentration below the toxic threshold.
Okay, these classical mechanisms, protecting the target, defeating the bullet, they really define the molecular arms race we see in the clinic. But recent research, especially looking at how bacterial communities survive, suggests this isn't the whole story, right?
Absolutely not. Not all interactions fit neatly within this simple bullet target model. The complexity, especially in real-world infections, demands a conceptual expansion.
We need to think bigger than just one cell versus one drug molecule.
We have to move beyond that. Recent work has introduced frameworks that operate at the population level or even the whole ecosystem level, revealing that survival is often a group effort.
And the first of these newer concepts is kin selection. That sounds like something from animal behavior studies.
It is, but it applies powerfully here too. Kin selection in microbes is fascinating. In this scenario, you might see a small number of resistant bacteria acting almost altruistically.
How so?
Well, they might be producing an enzyme, like a galactamase, that diffuses outside the cell and destroys the antibiotic in the immediate environment.
Protecting their neighbors.
Exactly. The key is that this action benefits all the nearby cells, including the highly antibiotic-sensitive ones, which are often their genetically close relatives, their kin.
So the resistant bacteria pay a metabolic cost. It takes energy to make that enzyme, but the sensitive majority gets to survive because the local environment is cleaned up.
Precisely. By protecting the sensitive cells nearby, The resistant few ensure the survival of the whole local population, the whole lineage, under antibiotic attack. Even if most of the colony is susceptible, the population survives and can keep propagating, potentially passing on those rare resistance genes.
It shifts the unit of survival from the individual cell to the local group.
Exactly. The focus shifts.
And the second concept goes even broader, moving beyond just kind to the whole community. Biofilm consortia.
Right. Biofilms, as we mentioned, are often incredibly complex communities. They're usually made up of diverse species, not just close genetic relatives.
A mixed microbial city.
A very good analogy. In these complex consortia, the protection against antibiotics, whether it's from the physical structure of the biofilm matrix itself or from drug destruction happening near the surface, that protection is effectively offered to all the community members living inside.
Regardless of their genetic relationship.
Regardless. The physical barrier of the biofilm matrix is a shared collective defense.
So the matrix itself acts like this massive sponge, soaking up or slowing down the drug, protecting everyone inside.
Yes, it binds and neutralizes drugs, slows diffusion right down, and just generally protects the entire community structure. This really demands that we adopt a full system-level view. We have to see the bacterial community as an integrated unit fighting the drug, not just a bag of individual cells.
We need to account for ecology, physics, community interactions, not just genetics.
Absolutely.
So what does this all mean? It means the problem is incredibly complex. It's going to require microbiologists, ecologists, doctors, policymakers, the public, everyone working together.
It has to be a concerted effort.
And crucially, before we even talk more about clinical solutions, we have to get a handle on the environmental source of all this ancient power that resistum existing outside of human medicine. Because that's where the story starts, and maybe where the solutions need to start too.
Yeah, if we accept that resistance is ancient, billions of years old, and that evolution has been honing these defense systems for eons, then the next logical question is, where are the clinically relevant genes coming from today?
Where are the new threats emerging from?
And our sources strongly suggest the most likely origin point for novel, clinically important resistance genes is that vast environmental antibiotic resistum: soil, water, sediments.
Okay, so this shifts the focus away from just treatment failure in the hospital and towards the fundamental ecological role these molecules play in nature. What's the leading thinking now on what antibiotics and resistance genes are actually doing out there in the wild?
The hypothesis is pretty groundbreaking, really.
Ah.
It suggests that the main functional role of many compounds we call antibiotics and their corresponding resistance genes in natural ecosystems is often primarily signaling or regulatory.
Signaling, regulatory, not just warfare.
Not primarily, perhaps. They might not be strictly lethal weapons or defenses in the way we think of them clinically. They could be communication molecules.
So a compound like penicillin or tetracycline found naturally in the soil, it might actually be used by bacteria there to communicate complex information, like population density or nutrient availability.
That's the idea. They could be information molecules helping to structure the microbial community, mediate interactions, maybe trigger developmental processes like sporulation or biofilm formation.
And how do we know this isn't just speculation? Is there evidence for this dual role?
Yes. Confirmation comes partly from observing that many antibiotics, when present at very low sub-inhibitory concentrations too low to kill, can have other biological effects.
Like what?
For instance, they can modulate host immune responses or trigger specific gene expression pathways in bacteria that have nothing to do with survival under lethal attack. This suggests a broader biological role than just simple chemical warfare.
Okay, and we see clear evidence of this environmental origin when we trace the lineage of some modern resistance genes, especially the ones that fight our purely synthetic drugs.
Absolutely. Take the quinolone class of antibiotics drugs like ciprofloxacin. These are entirely synthetic. They didn't exist in nature before we chemists made them in the lab.
Right.
They work by inhibiting bacterial enzymes called DNA gyrase and topoisomerase, which are essential for managing the coiling and uncoiling of bacterial DNA during replication. Yet resistance to these synthetic drugs is often caused by genes called qRNR genes. And where did bacteria get qRNR genes? They acquired them from aquatic bacteria, living in environments completely separate from clinical settings.
So that aquatic bacterium wasn't fighting Cipro back in the day. What was the original job of that qRNR gene then?
It seems to be a perfect case of what scientists call functional pre-adaptation, or maybe repurposing, Researchers looked into the original function of one of the Q&R molecules, a protein called MFPA. Its original job seems to have been providing DNA topological assistance. Basically, it helped maintain the highly condensed structure of the bacterial chromosome and prevented unwanted tangling or supercoiling, maybe during periods of stress or dormancy when the environmental bacteria weren't actively dividing.
So it had this sort of generic structural maintenance job for DNA, and then when that gene happened to get transferred into a pathogen, it serendipitously also provided protection against a synthetic drug that attacks DNA structure.
Exactly. It had a useful function just waiting for the right context. It really highlights this continuous flow of genetic information across boundaries, which brings us to the absolutely critical cycle of amplification and release.
Bacteria just don't respect our neat ecological compartments: soil, water, animals, humans.
It's one large, continuous genetic exchange network. A giant soup, as you said earlier.
Yeah.
So when these environmental resistance genes, even if they're rare out there, happen to enter the human or animal microbiota, the immense communities of mostly beneficial bacteria living in and on us,
Then what happens?
then the incredibly strong selective pressure exerted by our routine use of antibiotics immediately leads to the explosive amplification and horizontal dissemination of those specific genes throughout that host-associated bacterial community.
So human and animal bodies, and maybe farms, act as these massive evolutionary accelerators, concentrating resistance genes way beyond their natural levels in the environment.
Precisely. They become hotspots for resistance evolution, and the reverse flow is equally problematic and maybe less appreciated.
The flow back out.
Yes. Our massive use of antibiotics leads to the continuous release of both the antibiotics themselves, often unmetabolized, and this huge pool of pre-selected amplified resistance genes from human and animal compartments via hospital wastewater, municipal sewage, animal manure, agricultural runoff, straight back into the environment.
And our standard water and waste treatment plants, they don't always remove the drugs or the resistant bacteria or the genes themselves.
Often they are not sufficient, no. Sewage treatment, for example, reduces bacterial load, but often doesn't fully sterilize the effluent. So we end up discharging residual drugs and high concentrations of resistant bacteria and free-floating resistance genes back into rivers, lakes, soil, and potentially groundwater.
It's a constant feedback loop. We pollute the environment with drugs and resistance, which then makes the environmental pool even more dangerous for future acquisition.
Exactly, and this constant cycling generates a whole series of really urgent, unanswered ecological questions that we need to get a handle on.
Okay, let's synthesize those pressing questions our sources raise. First, what happens when all these different drugs we release mix together out there?
Right. We need to know, does this resulting multi-drug cocktail, this chemical soup in the environment, does it accelerate evolution towards even broader multi-class resistance?
In the lab, we usually study one drug at a time, but the real world is a messy mixture.
Good point. Second big question.
The presence of sublethal concentrations of antibiotics. These are traces of drugs that don't kill the bacteria outright, but definitely stress them out. What are the specific consequences of that constant low-level pressure? It might strongly favor resistance mechanisms like efflux pumps, which are particularly good at dealing with low drug concentrations.
Okay, and what about other environmental stresses, things we don't usually link directly to antibiotic resistance?
Think about things like UV radiation from sunlight, or maybe heavy metal pollution or certain industrial chemicals. These stresses often trigger what's called the SOS response in bacteria.
The SOS response, like an emergency mode. Exactly, it's the bacterial emergency repair system. When the cell senses significant DNA damage, it triggers the SOS response, which, among other things, makes the bacteria enter a hypermutable state. They make more mistakes copying their DNA, and crucially, they increase their rate of horizontal gene transfer.
So sunlight or pollution could actually make bacteria more likely to share genes, including resistance genes.
That's the concern. It could accelerate the entire engine of resistance evolution and spread in the environment.
Wow.
And all of that leads directly back to our own safety. What are the actual chances for these concentrated amplified resistance genes, now potentially mobilized by environmental stress, to re-enter the human and animal food chain?
How?
Through crops irrigated with contaminated water, perhaps, or through eating fish or shellfish from polluted waters. Getting a handle on these ecological pathways and risks is absolutely essential if we want to build strategies that can effectively preserve the power of the antibiotics we have left.
Okay, so the microbial world has made it abundantly clear. We can't win this just by searching for one silver bullet after another in the same old places using the same old strategies.
No, that approach has clearly hit diminishing returns. We need systemic change in how we find new drugs, how we target them, and crucially, how we protect the ones we still have.
So the path forward involves both innovation and stringent prevention. Let's start with innovation. The search for novel antimicrobials has to radically move beyond the traditional hunting grounds, right?
Absolutely. As we discussed, the soil actinomyces bacteria fueled the golden era. But digging deeper there, even with fancy new high-throughput screening, just hasn't yielded any truly novel drug classes for decades.
So where are researchers looking now? What are the new ecological niches?
They're exploring much more diverse environments. This includes the vast, largely untapped marine environment, oceans, deep sea vents, marine sediments. They're also looking at borrowing compounds, particularly antimicrobial peptides, from animals and plants that have evolved their own sophisticated defense systems over millennia.
And you mentioned accessing the uncultivated microbes. That sounded like a potential game changer.
It really is.
Yeah.
Historically, we could only find antibiotics produced by microbes we could actually grow in the petri dish in the lab.
Which is a tiny fraction of what's out there.
Estimated to be less than 1% of all bacterial diversity. Metagenomics changes that completely.
How?
It allows scientists to extract all the DNA directly from an environmental sample, say a scoop of soil or a sample of seawater, or even from the human gut microbiome. Then they can sequence that collected DNA and look for genes involved in producing potential antibiotics or interesting resistance mechanisms without ever having to culture the individual organisms.
So it opens up that vast 99% of previously inaccessible microbial life for drug discovery.
Exactly. It's a huge expansion of the search space, allowing us to find entirely new chemistries and biological activities.
And alongside exploring nature, there's also a return to the purely synthetic approach, the one Ehrlich pioneered.
Yes, that route is becoming much more prominent again. Instead of waiting for nature to provide a lead compound, chemists design and synthesize molecules from scratch, often guided by detailed knowledge of specific bacterial targets.
Made to order, essentially.
Right. This is particularly effective now that we can identify very specific molecular targets within the bacterial cell. They're essential for the bacterium, but ideally absent or very different in human cells. We can design drugs aimed precisely at those unique targets.
Some strategies even involve hybrid drugs.
Yes, some advanced strategies involve engineering drugs to hit two essential bacterial targets simultaneously, like a rifamycin quinolone hybrid antibiotic. The idea is that it's much harder for the bacterium to develop resistance to two different attacks at the same time.
Okay, this brings us to a really fundamental shift in strategy, redefining the magic bullet itself, moving beyond just hitting broad processes like building cell walls or making proteins.
Yes, towards incredibly precise molecular targets, guided by all the genomic information we now have. The vast majority of our current antibiotics hit maybe 5 or 6 major cellular processes.
That's a pretty small target list.
It is. But with thousands of bacterial genomes sequenced, we can now potentially implement Ehrlich's idea in a much more sophisticated way. We can define essential targets at a much finer molecular level, targets that bacteria hopefully haven't evolved common defenses against yet.
And one of the most exciting new paradigms here is a big conceptual leap, targeting virulence rather than just viability.
This is critical because it fundamentally changes the evolutionary pressure. Instead of trying to kill the pathogen outright, which forces maximum evolutionary pressure for immediate, high-level resistance.
You try to disarm it instead.
Exactly. You target the traits that make it pathogenic, the things that cause disease, rather than the things essential for its basic survival. The idea is that this might allow the host's own immune system to clear the less aggressive infection more easily without creating that intense kill or be killed selection pressure.
Can you give some specific examples of targeting virulence?
A prime example is developing inhibitors for the type 3 secretion system, or T3SS.
What's that?
Think of the T3SS as a tiny molecular syringe that many important gram-negative pathogens, like Salmonella or Pseudomonas, use to inject toxins directly into our host cells.
Oh.
If you can develop a drug that blocks that syringe, the bacteria might still be alive, but they can't effectively inject their toxins, making them much less dangerous.
So you jam the weapon.
You jam the weapon.
Yeah.
Another example is inhibiting QSEC-mediated activation of virulence genes.
QSAC, that's a sensor system.
Right. QSEC is a sensor on the bacterial surface. It detects stress hormones from the host, like adrenaline, and also signals from other bacteria. When it gets triggered, it tells the pathogen to switch on a whole battery of virulence genes, launching a full-scale attack.
So inhibiting QSEC is like cutting the communication line.
Exactly. Developing small molecules that block QSEC basically interrupts that signal. It tells the pathogen army to stand down, significantly reducing its ability to cause severe disease.
Taking away the coordination and the toxin delivery. The immune system might then have a much better shot at clearing the infection without needing a drug that kills the bug directly.
That's the hope. And another really interesting strategy in this vein is drug repurposing.
Finding new jobs for old drugs.
Exactly. Identifying existing drugs already approved for human use for something else entirely that just happen to also inhibit a bacterial virulence factor.
Yeah.
The huge advantage is speed and cost, because these drugs have already passed extensive safety trials.
Do we have a concrete example of that kind of lucky overlap?
Yes, the sources mention a compound called BPH 652. It was originally tested as a potential cholesterol-lowering drug in humans
Okay.
because it inhibits an enzyme in our cholesterol synthesis pathway called squalene synthase. Intriguingly, researchers discovered that BPH 652 also powerfully inhibits a related enzyme in Staphylococcus aureus, the bug that causes MRSA infections. This enzyme is called dehydrosqualene synthase.
And what does that enzyme do in Staph aureus?
It's crucial for making the characteristic golden yellow pigments, the carotenoids, that give golden staph its name.
And those pigments aren't just for looks.
Not at all. They are critical virulence factors. These pigments act like antioxidants, protecting the bacteria from the oxidative burst defenses used by our immune cells, like neutrophils.
Ah, so the pigment is like sunscreen against the immune system?
A very good analogy.
Yeah.
By inhibiting dehydrosqualene synthase with BPH 652, the bacteria lose their pigment, they become white, and they become much more vulnerable to being killed by the host immune system's reactive oxygen species.
So BPH 652 could be repurposed as an antivirulence drug to help control MRSA without directly killing the bacteria itself.
That's the potential, a really promising avenue.
That's a huge return on investment, finding a new use for a drug already proven safe. What other kinds of new molecular targets are researchers looking at beyond virulence?
Well, the field is exploring pathways that are essential only to bacteria, hopefully minimizing side effects in humans. Things like specific enzymes involved in bacterial fatty acid biosynthesis, the machinery controlling cell division, other types of bacterial communication like different quorum sensing systems, and various bacterial two-component signal transduction systems beyond QSEC.
The key is finding targets unique to bacteria, maximizing impact there while minimizing cross-reactivity with us.
That's the goal. Maximum specificity.
And instead of just trying to beat the bug with new bullets, we can also try to target the resistance mechanisms themselves, right? To make older drugs work again.
Yes. This is a crucial complementary strategy, essentially turning the tables on the bacteria's defenses. We can develop highly specific inhibitors aimed at key resistance mechanisms.
Oh, like
Inhibitors for the different classes of active antices, the enzymes that destroy penicillin, or small molecules designed to bind to and disable the efflux pumps, those bilge pumps that kick antibiotics out.
So you block the pump, and the antibiotic can build up inside again.
Exactly. If we can successfully inhibit the resistance mechanism, we might be able to bring older, safer, cheaper antibiotics back into clinical use, effectively extending their therapeutic lifespan.
Okay, and finally, there's the idea of combination therapies, not just mixing two antibiotics, but some really unconventional, almost ecologically minded approaches.
Yeah, the combination therapy concept is expanding rapidly to include biological agents too. For instance, combining antibiotics with specific bacteriophages viruses that infect bacteria has shown real promise.
Phage therapy.
Right, especially against cuff infections like those embedded in biofilms. The phage can sometimes disrupt the biofilm structure or kill cells deep inside, making the conventional antibiotics much more effective when used together.
And the example of protecting our own beneficial gut microbes during therapy, that sounds maybe the most fascinating in terms of containing future resistance evolution.
It's the ultimate expression of trying to be precise and contain the fallout. Think about when a patient gets intravenous ampicillin. A small amount of the drug inevitably gets excreted into the gut.
Where it's not needed for the primary infection.
Right, but where it can wreak havoc on the beneficial gut microbiota, killing off good bacteria, and powerfully selecting for ampicillin resistance among the survivors in the gut flora.
Okay.
So researchers have explored giving the patient an engineered recombinant volactamase enzyme orally at the same time they receive the IV ampicillin.
Wait, you're giving an enzyme specifically to destroy the antibiotic. That sounds counterintuitive.
Yes, but only in the gut, where the drug is doing ecological damage rather than treating the systemic infection. This specialized enzyme isn't absorbed, it stays in the gut and neutralizes the ampicillin there.
Temporarily shielding the good gut bacteria.
Exactly, preventing the therapy from accidentally selecting for resistance in our own commensal flora. This could significantly limit the creation and subsequent shedding of new resistance genes back into the environment or transmission to other people.
That is next-level precision thinking. Amazing. But none of these incredibly clever, innovative strategies will matter much if we don't drastically improve prevention and regulation on the other side of the equation.
That's the hard truth. That's the final and perhaps most difficult component of the path forward.
And the core issue here comes down to consumption, doesn't it? Especially indiscriminate consumption.
The data is just unequivocal. The level of antibiotic-resistant infections in a country or region correlates strongly and directly with the total amount of antibiotics consumed there.
More use equals more resistance.
It's a clear relationship. Moreover, another critical factor is public knowledge. Studies show that a general lack of awareness among the public about what antibiotic resistance is and how it happens positively correlates with a higher prevalence of resistance.
Which points to a massive failure in education, but also in diagnosis and policy.
Absolutely. We desperately need targeted patient education to prevent inappropriate use, like people demanding or doctors prescribing antibiotics for viral infections like the common cold, where they are completely useless.
But it's not just patients. Professional practice needs reform, too. You mentioned empirical prescribing.
Yes, the vast majority of antibiotic prescriptions worldwide are still empirical, meaning the doctor prescribes based on their best guess about the likely pathogen and its probable sensitivity pattern, often opting for broad-spectrum drugs just in case.
Why is that still the default? Why not test first?
It largely comes down to time and logistics. Properly identifying the specific pathogen and then testing its sensitivity to a range of different antibiotics doing an AVR profile. An antibiogram traditionally takes time, often 24 to 48 hours, or even longer.
And the patient is sick now.
Exactly. The patient is suffering, and delaying treatment can be dangerous. So doctors often prescribe a broad-spectrum antibiotic immediately to cover the most likely culprits. The development and widespread adoption of rapid point-of-care ABR profiling tests that could give results in, say, under an hour, would be a total game changer.
Allowing doctors to choose the most effective narrow-spectrum drug right from the start.
Precisely. Avoiding that unnecessary selection pressure from the shotgun approach.
In many parts of the world, this whole problem is made much, much worse by cultural and economic factors too, isn't it?
Hugely so. In countries where the sale of antibiotics is inadequately regulated, where you can just buy them over the counter without a prescription.
Self-medication becomes common.
Yes. Personal decisions about buying and using antibiotics are often driven by cost, convenience, or self-diagnosis based on past experience or advice from friends, not a proper medical assessment. This kind of self-medication lacks all the basic attributes needed for successful therapy: proper diagnosis, correct dosage, taking the full course, monitoring for side effects.
And it massively contributes to resistance by constantly exposing bacteria to inappropriate drugs or sublethal doses.
Immensely. It's a huge driver of resistance globally.
Okay, moving beyond human health, the agricultural sector. This seems like maybe the largest continuous reservoir of selection pressure outside of hospitals.
It's definitely a major contributor. Domesticated animals, like humans, absolutely require antibiotic therapy when they get bacterial infections. That's not the main issue.
The issue is non-therapeutic use.
Exactly. For decades, antibiotics have been widely used in agriculture, aquaculture, even horticulture, not just for treating sick animals, but for growth promotion, helping animals gain weight faster on less feed, and for routine prophylaxis, preventing anticipated infections in crowded conditions.
So, massive amounts of antibiotics used continuously on healthy animals.
Yes. This creates a gigantic, persistent selection reservoir, constantly breeding and amplifying resistant organisms and genes, which then contaminate meat products, shed into manure, and leach into the environment.
So what's the model for fixing this? Has anyone successfully tackled agricultural overuse?
The experience of the Scandinavian countries offers the most encouraging, actionable example. They started implementing comprehensive programs decades ago.
What did they do?
They focused on multiple fronts: improving farm hygiene and management to prevent disease outbreaks in the first place, establishing strict guidelines for proper antimicrobial use only when necessary, and, critically, They withdrew antibiotic growth promoters from use in food animal production way back in the 1980s and 90s, long before the rest of the EU did.
And did it work? Did withdrawing growth promoters lead to less resistance?
Unequivocally, yes. These early and aggressive measures resulted in a significant reduction in the overall use of antimicrobials in livestock in those countries, and consequently, they have a much more favorable situation regarding antimicrobial resistance levels today compared to many other regions where these policies were implemented much later or are still lagging.
So it proves it can be done.
It does. The EU-wide ban on all antibiotic growth promoters, which finally came into effect in 2006, followed directly from the success seen in Scandinavia and recognized the absolute necessity of limiting this massive source of resistance dissemination. So as we wrap up this deep dive, we really have to come back to the central tension driving this whole crisis. The microbial world, with its almost 4 billion years of evolutionary history, has accumulated this just enormous, incredibly diverse genetic library of metabolic tricks and protective mechanisms.
And those mechanisms are ready to be mobilized?
They are standing ready, poised to be mobilized against any massive, indiscriminate antibiotic assault we launch. Our fundamental failure, perhaps, has been failing to respect the deep, ancient ecology of resistance. We treated it like a simple engineering problem when it's actually a complex ecological and evolutionary one.
So the key takeaway for you, the listener, isn't despair, hopefully, but a call to action. We need that radical precision in targeting pathogens, new drugs, new strategies like antivirulence. But just as important, maybe more so, we have to immediately limit the indiscriminate use of the precious antibiotics we still have.
Everywhere. In the hospital, on the farm, in the community. We have to stop accelerating the emergence of novel resistance mechanisms through careless overuse.
And investigating the microbial world all around us, that environmental resistome, the genetics of ancient resistance that gives us the foundational knowledge we need.
Not just for finding new drugs, but for building crucial early warning systems, for spotting the next resistance threat before it hits the clinic, and for designing truly preemptive, preventive measures that can sustain the efficacy of chemotherapy for future generations.
It's a powerful and maybe humbling lesson in evolutionary ecology. We know now that resistance genes existed for billions of years before we ever discovered penicillin.
Long before.
So if their original role wasn't primarily to fight our modern drugs, but possibly, as you suggested, to act as sophisticated signaling or regulatory molecules in natural ecosystems, what other unseen functions do these ubiquitous genes play in the global microbial world?
And what are the consequences of us disrupting those ancient functions through our widespread indiscriminate use of these compounds?
That is the thought we want to leave you with today. The molecules we call antibiotics might just be part of the language of the microbial world.
And maybe we need to learn how to listen better and be more careful about interrupting that ancient conversation before we lose the power of every word we have left.
Until next time, keep digging deeper.